All right, so good morning, everybody. It's a pretty small crowd, so please, I do want this to be a little bit more interactive. That's kind of one of the things when it's a smaller crowd. Do feel free to talk, raise your hand. When it's this small, we've got time to do that, so please feel free. First of all, my name is Eric Evans. I work at Goodblend. We're one of the 3 medical cannabis licenses in Texas under the TCUP program. I'm their director of manufacturing, and I have been with the company for 7 years, going on 8 now. I've been here in Texas since the medical cannabis program started, was involved in the licensure and application process out here, and have been here ever since. A little bit about myself and my background. I'm originally from Atlanta, Georgia, so I'm not a Texan. I'm one of those that says, got here as fast as I could. And cannabis actually brought me out here, so that's actually pretty exciting too, since it's a pretty small market even still, especially on the medical side. I went to Kennesaw State University. My degree is in a Bachelor of Science in Chemistry with a concentration in pharmaceutical sciences. After that, I went to the Mercer College of University School of Pharmacy, did that for 1.5 years. And then that was when I decided to get out of the track that I was going on, which was towards becoming a pharmacist and get into the cannabis industry. It was a very scary moment in my life to do that. It was a very solid, stable career trajectory that I was on, and to kind of pump the brakes and be like, no, I think I can still make a good decision for myself professionally and personally in my connection to this medicine. So I made the decision to get out, and I started on the front lines as an hourly frontline worker with what was at the time Certera, which is still how we operate in Florida. That's that frontline formulations job I had. While I was there, I was doing pretty much everything you've heard of: tinctures, gummies, vapes. I was on the front lines making those things in the day-to-day. So I've been all the way from the front lines and worked my way all the way up to a supervisor director type position that I'm in now. In between those, like I said, I came out here and I was involved in the licensure and application process with DPS. So I've been working very closely with the DPS here in Texas for the last 7 years. A very close relationship with them. And if y'all have any questions about that and what that's like, I'll be happy to answer those at the end. It's interesting and it's different in every state for every regulatory body. I was also heavily involved in R&D. So that's a lot of what we're going to be talking about today is how do you develop products? How do you make good products? And what does that look like? How do we start making that a consistent thing throughout the whole industry for all of us, especially here in Texas? That's my background. This is one of those things that everybody hates, but I like doing these things because it makes you start thinking, and it's early in the morning. So I'm not going to go to the next slide until you take a couple seconds. What do you think personally? And I'm not going to ask you, I'm not going to make anybody raise their hands because I'm not that guy either. Don't tap the mic. But yeah, what do you think is the most important thing in this industry going forward? What is the one thing that we really, really have to do? This whole presentation is really my take on what that is. So I'm going to give you a second, think about it. I'm going to show you what I think it is. This is what I think it is. It's patient trust. If we lose this, if we damage this, if we have something that causes damage to this relationship, we're done. And this is a nationwide problem right now because, and we'll get into the details of this, but there's a lot of confusion that's happening purposefully and not purposefully. I think there's some good people that are trying to do the best that they can, but are causing confusion. And then there are people who are actually actively trying to take advantage of loopholes and use the confusion of the chemistry to take advantage of trying to sell things before they really are regulated properly. So it's not to say that I'm against where we are at right now. I just think that there's some things that we could do better to make sure that we are maintaining patient trust. And we're going to go into those things. So first, we're going to go into the state of things. Y'all have been here for a couple of days now. I'm sure y'all have heard all of this, but just the quick overview of the program. There's sort of 2 operating rule sets right now within the state of Texas. We have the Farm Bill rules, which is the hemp companies and everybody that is operating under that rule set. And then you have the TCUP program, which is only 3 licensees right now and some pretty limited operations and pretty strict regulations there. It's loose in some ways and strict in others. We'll talk about that as we go. Y'all can look through this, but this is sort of just the state of things, right? We've got a limit of THC that's a little bit different, but I'm sure y'all have heard this. A limit by weight's not a real limit. We'll talk about that more, but 0.31%, that's not a limit. You can raise the weight, you can raise the dose. A cookie is very, very heavy. A bottle of water is very, very heavy. You can dose that to the sky because it's heavy. And this is a weight limit. So you just raise your weight, you can raise your dose. So whatever they were trying to do from a regulatory standpoint by limiting the amount of THC by 1%, don't know. Not sure. It's not the way. We've got to figure out a way to coach our regulators away from that way of thinking, because it's not a limit. And if that's what they're trying to do, we need to coach them in such a way that it's not going to be detrimental to patients, that they don't make a decision that says, oh, well, 5 milligrams per dose. Whoa, now they've stepped on our toes and we can't treat patients properly, right? So that is something we got to be careful about moving forward. Water real quick. Want to talk about the drop in CBD value. I'm not going to spend too long on that because I can get on a little soapbox of it, but it is one of the reasons that we're starting to see all of these different variants coming out in isolate form from these manufacturers. You had a ton of CBD being manufactured through the Farm Bill. And what that did is, unfortunately, it led to a huge drop in the price of CBD. So all of these farmers that were wanting a really big return on all of this hemp investment suddenly were getting pennies on what they were expecting dollars on. And it is a big problem. So what they started to do, and I'm not saying this is bad, I actually think this is the way forward, is they started turning that CBD into more profitable molecules. CBN, THCV, all of these more trace cannabinoids can be made from high quantities of CBD. So you're starting to see those things come out. I'll get into this later too, but you're starting to see other things like HHC come out. I'll talk specifically about HHC later and why I don't like that molecule. We have THCA flower coming in from out of state being sold under the Farm Bill. Not going to spend a lot of time talking about that. It just is the state of things. You can go to just about any hemp shop and buy weed, regular potent weed. It may be old. It may have been tested 6 months ago. It may be fresh, depending on the supply chain that person has set up. It is very variable from shop to shop because there's not a whole lot of regulation regarding how those things are brought in and how they're kept. With us, again, you're not seeing much difference here. So under the medical program, we have a 1% limit by weight. All of our APIs are supposed to be plant-derived, which is technically true of everybody, and that is true, how we're all operating, right? We don't have any flower sales yet. We're not selling any form of inhalables under the medical program. It's strictly edibles. And we are constantly inspected by the DPS. Again, I'm not advocating for saying these things are good or bad at this point. I'm just letting you know where the state of things are. The other thing is that we have to batch test every single batch in the medical market. That one I will advocate for right now. Everybody should be testing every batch that you're making every single time. It's just imperative because you can't be perfect every time, and that's the only way that you'll know. Later on, I'm also going to talk about some of the things that you can do to vet your third-party labs to make sure that what they're telling you isn't bogus. So how do we all be good operators? This is how we cultivate that patient trust, right? If we're all doing the right things and we're all operating in a good, trustable, respectable way, then we can start building these bridges. And now patients can rely on us. They can rely on our labels. They can rely on what we say. And when they call us and they ask us questions, we can build a sense of positive community around this relationship. Because if we start building a sense of negative community, we're going to have an uphill battle, right? Because we are still in the state where we're not totally legalized. We have got to get public buy-in. We have got to get people on every level of our society here bought into this. So we cannot be bad operators. So we're going to talk about how do we avoid being that? How do we be good operators? The patient trust side is the left, and then the thing on the right is the acts that we're going to do. So the rest of the presentation, we're going to go basically through the things on the right and talk about how we can do these acts, and the things on the left, yeah, making sure that my directions are correct here, are the patient trust things. So you got to make high quality products. If you're not making them, you have to make sure that you're selling high quality products. If you've got something on the shelf that is, for lack of a better word, garbage, and it goes to a patient and it's their first time experiencing cannabis, we've lost somebody. And not only have we lost that person, we've lost every single person that they talk to if they have a bad experience. Oh, I tried that cannabis and I don't know what everybody's talking about. That was horrible, right? So we got to make sure that people are getting good experiences when they have these products, because any individual patient has a network of people that they're talking to, and that's really important. One way we can do that is a low degree of variance, and I'll get in much more detail on that. I've got a lot of slides on that one. And another way is truthful labeling. So you've got to make sure if you say 10 milligrams on the label, it's 10 milligrams. It's not 7, it's not 15, right? And it's consistent every single time because patients are trying to treat this a little bit like they treat their pharmaceuticals or their dietary supplements, right? It's going in the pill counters a lot of times with these edibles. I'm taking this twice a day or 3 times a day to try and manage this condition. It's a very different way of thinking about it, right? We got to be really consistent under those guidelines. I'm also going to talk to you about how to vet a third party. I already mentioned that. You can also use those same principles when we get to that to vet your supply chains, right? If you are buying a hemp-derived THCA or THCV, CBG, whatever it is, right? There's a set of questions you need to be asking those suppliers regarding that input that they're selling you, because they're going to tell you a lot of things because they have to, because of the legal structure that we're currently in. But there's a couple of questions that you can ask them to kind of get to the real truth of what they're selling, to make sure that what they're selling you is clean, high quality, and consistent, right? And that's how we're going to build community trust. For the Harry Potter fans, I threw in constant vigilance from Mad-Eye Moody in here, but that is one of the things that we do, right? You have to be constantly vigilant on these things. You can't ever pretend that you've figured it out and that you're on top of it and that every single time you're right. You can't. The second you do that, that's when you're going to send something out the door and it's going to be outside of spec, right? Like you've got to check every time. It's really important. And there's some things that you can do regarding analytical chemistry to check for that. I'm going to teach you some of those things in here today. And we're going to go over those basics. All right, let's go into high quality products. Low degree of variance. This is one of my soapboxes. I will try not to stay on it too long. So the FDA and the FTC. The FDA is typically the one who is going to be regulating your pharmaceutical products, and the FTC is typically the one who's going to be regulating things like dietary supplements, things that you're buying over the counter. A lot of people don't know this, but when you buy any pharmaceutical or any dietary supplement, there is a degree of variance that is allowed on that product, okay? So if you are buying a 100 milligram product, it's going to allow a range of variance from that 100 milligrams. Be it down to 90, up to 110. It varies based on different products. Highly toxic drugs, drugs that are, if you take a little bit too much, it's going to really hurt you. Those have a much tighter variance. They might only allow 1% or 0.5% variance from your label claim, because if you take too much, it's going to cause problems, right? Whereas well-tolerated drugs, which is where we operate, we are in the cannabis space. This is a very well-tolerated drug. We've seen almost no toxicity in many, many varying levels, other than a bad experience. Those allow for higher variances. So the highest variance you typically see is within dietary supplements. That's up to 20%. And in most of your pharmaceuticals, it's down to 10%. All right, let's go through an example. So for a low degree of variance, if we're talking about a 15% variance, right? So this is somewhere in the middle, right? This is what a lot of states within the cannabis industry have chosen as the degree of variance that they accept, 15%. So if you have a 100 milligram chocolate bar that you're selling and you are allowed a 15% variance, that means very simply that you could have 85 milligrams to 115 milligrams in a bar, and that would pass your label claim. What it's basically saying is that anything outside of that, now you're not within your label claim. Now, you saying it being a 100 milligram bar is not actually true. So you shouldn't say it's a 100 milligram bar. And the tighter that variance is, the harder it gets, right? The better you have to be. If it's a 2% variance you only allow yourself, you're probably going to end up failing a lot. You're going to have to reformulate throughout batches. That's why you have to be, there's a delicate balance here, right? Like you can't be perfect, but you should hold yourself to a high standard and say, okay, I'm going to hold a tight variance here. Let's look at a 5 milligram gummy. It gets a little harder when it's a very low dose, right? So 100 milligrams, you see that it's a pretty big range where you can mess up, right? Let's not say mess up. Pretty big range that you can formulate within and it still passes. For a 5 milligram gummy, it gets a little tighter, right? It's a 0.75 range that you can be off on because it's a smaller total. It's a smaller area that you can actually be off by. So the range for a passing gummy that's a 5 milligram label claim with a 15% variance could be as low as 4.25 milligrams per gummy up to 5.75 milligrams per gummy. This is why analytical testing is so important, and providing COAs to your patients when they ask for it is so, so important. I cannot tell you how many times I will get a phone call or an email from a patient that says, these hit a little bit harder this time, or this didn't hit quite as hard this time. What's going on? It's a couple of questions you can ask. Did you have anything on your stomach? And then I'm going to look up these test results. It could have been a little lower. It could have been a little higher. That's one of the first things I'm going to check. And then I have a very clear explanation that I can give to the patient, right? This dose is still within range, but it was a little bit lower, so you might want to take another half, something like that. It gives you more power to have that open conversation with the patient, and then they don't feel like you're doing something tricky to them, right? It felt a little different, but if you can't explain why, they might not trust you as much. And it's important to be able to explain those things to them. So, current takeaway from variants. Currently, neither, to my knowledge, the Farm Bill or TCUP within the actual bills have variants within them. This is not something the legislators caught and said that we need to be looking at. So the DPS has asked us to operate at 15%. They have created a rule, which is within their right to do. And we support that. 15% is good. We choose to operate at 10. We do not sell batches that go outside of a 10% variance for ourselves. As operators and as sellers and people who are manufacturing, that's up to you, right? Like, I wouldn't go outside of 20. 10's pretty tight, right? I advocate for somewhere within that range. It's really important that we all together, though, agree to start selling products that are within those specs. Because, again, we don't want somebody to pick it up and have something that's really, really hot. That's actually the worst example, right? If they take it and they don't really feel much, that's not the worst in the world. If they take something and they have a really bad experience and now they're turned off to cannabis, that's what none of us want, right? All right, and the other thing that thank you, I'm glad I have notes on here, hey. Products coming out of other states that a lot of times is what we're selling here in the hemp markets, you just gotta check, because those variances may not be the same, right? They may not exist like it is here. They may not actually have a variance stuck in their rules. So that could be one of the things that you're asking one of those suppliers that is selling you an active pharmaceutical, right? If you're buying your THCA or your CBD from somebody, you need to be asking, what's your variance when you're selling these products? What is it per gummy, right? Or per chocolate bar, or whatever it is, the product that you're buying. Questions on variance before we move on? Great, cool deal. All right, truthful labeling. There's more slides on this. This is more the introduction of what's to come. We are going to talk a good bit about how to vet your suppliers and how to vet your third-party labs, how to look at their standards, how to evaluate a curve. And I did see there's another talk after this one that's even more of a deep dive on that, which I'm super happy to see. I'm more going to be giving you the lay person. If you're not a chemist and you want to kind of know, I'm calling up a third-party lab and I don't really know what I need to ask. And these people are confusing me with big words. Because they will. It's an easy way to make you feel like you don't know what you're talking about. As a chemist, I'm telling you, it's easy, right? Like, I can just throw words at you and be like, all right, here's the price. What do you think? I'm going to tell you the things that you can cut through all of that and just get to the real meat of the bone, and you'll probably disarm them and they won't be ready for it. So, all right. I also want to say with labeling, with the FTC and the FDA coming, we don't know which one we're all going to be under eventually at this point. It's probably going to be the FTC, if I had to guess. I would go ahead and recommend get ahead of the labeling problem, right? Like, if you are creating labels, if you are purchasing labels, just go ahead and do it to the FTC's guidelines. That way you are not having to scramble whenever the government finally gets their shit together and we have legality here, right? Because you don't want to have to then suddenly replace everything on your shelves. Or now you've got a couple $1,000s of labels sitting on the shelf that you can't use because the FTC rules are now suddenly in effect. They're not that hard to follow. It's typically things like ingredients, order of ingredients, manufacturer address, things like that. So just go ahead and get ahead of those things. I highly, highly recommend it. It's going to save you money in the future, I guarantee you. Okay, so this is going to be my quick slide on actual manufacturing of cannabis products. I didn't want to get too into the nitty-gritty of this because this slide alone could be an entire presentation of itself. I do want to give you all an opportunity to ask questions, though, on this slide. So this is about how do you take your active pharmaceutical ingredient at this point, be it isolate, be it distillate, be it you've got raw flower that you've got to do something with. How do you take, or RSO, whatever point in the process you've got your cannabis at, how do you take that and put it into an edible product? Specifically talking about edibles for this first section. There's one thing that you've got to think about, and it's your product matrix. What are the components that are making up the product, and which parts of that can cannabis go into readily? Because remember, cannabis is a fat-loving molecule. It's an oil-loving molecule, right? So if you've got water at some point in your process, that ain't it. Don't put the cannabis in the water step, right? You need to put it in your oil-based step, wherever that is, whatever your matrix is. And it varies per what you're looking at. So let's go through a couple examples. If you're making gummies, be it gelatin, be it pectin, be it both, doesn't matter. That is a water-based product. There is a good chunk of water in most of your gummies that you are making. So you need to be strategic and look at the process. Where is your sugar? Where are your oils? Where are those parts of the process? And don't even think about it being a cannabis gummy for a second. Go online or wherever you're getting your recipe from and look up a good gummy recipe. Pretend it's not a cannabis recipe. Make that first. Get that first, right? Get that good. Get your texture happy. Get all that stuff happy. And then think about where you need to put in the cannabis, right? And it is not going to be the water step. That's your biggest indicator, right? If you've got a boiling pot of water, it's not there. It's probably over here with your sugars and your oils and your flavorings, okay? The next thing that we're going to look at product matrix wise is chocolate. It's very, very, very different from gummies. There's no water in chocolate. If you've got water in chocolate, you've got problems. And I tell you, if you do have water in problem, it's going to be a big nightmare for you. So do make sure your space is dry, and do make sure that you've got no water in your space for chocolate. For chocolate, it's a very different matrix. You cannot just burr. Do y'all know what burring is, by the way? It's like an immersion blender or a homogenizer, essentially. You can't do that to chocolate because you're going to whip air into it, and that's going to cause problems. With gummies, if you whip the air into it, that's not a problem. You can deal with that. There's other ways to deal with that, right? But with chocolate, it's difficult. So you have to look at it differently. It's a totally different monster, right? And you have to treat every new product like that. Every time you're looking at a new matrix, it's a new way to infuse the cannabis. And you've got to make sure that it's consistent throughout the batch. So you've got to test it at multiple points, right? So you've got your big vat of chocolate. You can't just test the top and it comes out great. And you go, okay, good, we're done. Pour them all, let's go. You need to test the top, the middle, the bottom. You need to test several bars. Not every time, right? But when you're first validating your method to make sure that you're incorporating your medicine all the way through, you need to check multiple times to make sure that it's getting consistently throughout, that they're all within that tight variance, that every bar that you're pulling out is coming within that 10 to 20% that you've selected for yourself, right? Oil-based matrices, that's not even hard, right? Like if you're just putting cannabis into oil all day long, send it, you can do it. Put it in olive oil, put it in any fat-loving molecule, it's going to be really easy on you. So all those formulations are kind of low-hanging fruit, honestly, for us because of the molecule that we're working with. Water is the literal exact opposite. So it's actually kind of amazing that we see the mass proliferation of beverages that we do on the market right now, because putting cannabis into water is literally putting oil into water. It's kind of a tougher chemistry thing to do. Solubility-wise, you need to create an emulsion, or more of like a gravy or a roux for those who are the cooks in the room. That's literally essentially what they're doing when they're making these cannabis beverages, because you're having to grab an oil molecule and then the water molecule, and something has to hold those both together. And there's different ways you can do that, right? Like if you are making a gravy, you need your fats and your waters in there, and you have to mix them in certain ways. So water is tough. So if you really want to get into the water space, be prepared to do some heavy chemistry lifting. It's not light. The others before, a lot of it you can kind of figure out logically through trial and error and just doing the work yourself, right? Water's tough because we are literally putting oil in water. Yes. Like, do you mean flour? Like cannabis flour? Cannabis teas would be challenging, specifically because you might get the plant components coming out into the water that are water soluble, but you might not get very much of your actual cannabinoids into the water. Now, could you potentially do like more like a crystal light type thing, probably, where you've infused a powder to create a tea. That's something that could be done. But you're gonna have to do something chemistry-wise to get the oil to go into the water. And usually, like I said, it's usually like a long molecule that can hold on to one and the other. So it's doable, but you could control that tighter. Yeah, because you would know exactly how much, because you could test that, right? If you make something and you can mix it up 3 or 4 times, test all those, the tea bag, I promise you, is going to be different every time. How hot was the tea? How well did it infuse? And again, it's not water soluble, so not very much is going to go in. Yeah, absolutely. Any other questions before we keep trucking? Cool. Yeah, and so this is kind of what I was talking about a little bit. Homogeneity. I hate this word. I've hated it since college. Homogeneity is not it, but that was close. So we're going to go with that. You're making a homogeneous solution. We'll go with that. All right. So some of that is trial and error, right? Like I said, you make your gummy recipe, you get your textures and everything right. Now you infuse it. You need to test that several times, right? Like I said, you got to test the top, the middle, the bottom, and there's ways to make sure that moving forward, you don't have to do that every single time. You can test 1 or 2 gummies from your batch, 1 or 2 chocolate pieces from your batch, and validate that your process that you've put in is making consistent products. That requires, though, you've got to have a really good relationship with your third-party lab, or you've got to have some in-house testing. One of the two. We're going to get into analytical testing. Thought I deleted this slide last night. My bad. This is just us, though, so I will go over this slide. This is what we do, and this is how we validate. This is our internal analytical lab. If you do your own internal testing, you do need to externally validate. Let me say that again. If you do your own internal testing, you need to externally validate. Otherwise, you are the professor giving the exam and grading the exam and receiving the score on the exam. See any problems with that? Right, so you have to go and get third-party validation, right? So we do that via Emerald Scientific, and we do it through the DPS testing via a third-party lab to validate our own methods, right? So Emerald Scientific is one of the companies in the chemistry world that really got on the cannabis bandwagon early. They and Shimadzu and a couple others recognized that there was a need for high-quality chemistry to be happening within the cannabis industry, and there weren't suppliers willing to give us the chemicals that we needed because they all had federal contracts and they did not want to touch us. And I'm telling you, in the early days, getting a handful of cannabis standards was jumping through hoops. Nowadays, it's a lot easier, right? These companies that got on it in the early days really hold the space now, so you can trust them. Really, I do advocate for using Emerald Scientific to validate your testing labs. What they'll do is they will send you, depending on what you're testing. If you're testing chocolates, they'll send you a chocolate. If it's gummies, they'll send you gummies. And it is an unknown to you. You don't know how much is in that bar. They know, they have validated it, they have tested it. You run it against your method, you send back the results to them, and if it is within that variance that we just talked about, they will give you a validation that says your method is testing these correctly and it is creating a result that's within the range that we allow. So you do want to check for that. Yes. We are not required to, but it is one of those things that it's a little bit of a CYA, and it's a lot of a checking of yourself, right? Because it is very easy in the chemistry world as an analytical chemist to have one small error somewhere in a spreadsheet. You're off by 1 decimal place. I'm telling you, it can be something as simple as you're off by 1 decimal place. And you may not catch it. You may not catch it if you're not doing certain things, right? And by getting these external validations, you're knowing, and somebody else is telling you, no, you're right. And it's also good, right? Just from a self-knowledge standpoint, right? Like if you're testing something, you can think you're right all day long, but having the professor tell you, no, you're right, that's helpful, right? And in this instance, that's kind of what it is. It's kind of having somebody else say, no, you know what you're doing. You keep doing that and you can trust it. But like I said, constant vigilance, right? Like you can't ever sit on your laurels, right? You've constantly got to stay on top of this. You should be doing these validations every couple of years, right? And then the other way we validate it, like I said, the DPS will take our products and run it through another third party that they have validated. So that is another way that we know our products are coming in within a tight variance through our testing lab. All right, little humor. Please don't get mad. I do not have rights for anything Monty Python, so let's all pretend this slide wasn't here as we move on and chuckle at the Monty Python humor. These, wow, what is going on? There's supposed to be a third, and I'm hoping that I will click and it will slide in. We will see. So there are 3 questions that you can ask an analytical lab to sort of cut through all of the mess that I was talking about earlier. One of them, what is your procedure for a failed test? This is going to tell you a lot about the quality, the integrity, and the ethics of that lab. How they answer this question is going to tell you if they are going to give you the answers you want or the answers you don't, or if they don't care what you want, which is what you want. You want a lab that doesn't care. You want them giving you the truth. It's right or wrong, doesn't matter. Tell me so I can fix it, right? So if they have a failed test, the answer should be, well, first, we retest it. And if it's still the same, we reach out to you, and then we may take another sample and check the batch again. But after that check, if it fails again, you need to fail that batch. And if they say anything other than that, you need to be suspicious. If they take that second or third or fourth sample and retest that batch for you until it passes, that's lab shopping. It's lab shopping within the same lab, but that's lab shopping. And you can pick that out very quickly if they're willing to do that. And you can flat ask them. So if my batch fails 2 times, can I send you a 3rd sample? Could I send you a 4th sample? Would you be all right with that? And if it passed on that 4th sample, would you pass the whole batch? If they say yes, you run to the hills. Because that means they are just giving you a pass and test result. Because they're charging you every time. Yeah, I'll take another sample, send it on, buddy. $500 a pop, let's go. That's a great business model. So that's a good question to ask. Failed test. The next one, how often are you running a check standard? That's basically the internal version of doing that external check, right? So in the Emerald example I gave y'all, they send you a 100 milligram bar, or excuse me, an unknown bar, right? It's maybe 95. You test that, you run it, you send it back. A check standard is very similar. You'll buy these from a company like Emerald, and it's a known amount. 50 parts per million every time. If you run it, it's 50 parts per million. You should be running those pretty frequently, especially if you're a high volume lab. If this is a lab that is churning through a lot of samples, they need to be running that known probably every 100 samples or so that they are checking and making sure every time I run 50 parts per million, it says 50 parts per million. It sounds trivial, but it is just literally making sure that the detector in the machine is telling you the right thing. And if they're not doing that, it could have changed. Something could have happened to their column, something could have happened to the detector, and now it's off by 5 milligrams for every result. And they're not catching it because they're not running that check standard. If you run a 50 parts per milligram check standard and it comes out at 60, now you know you've got a problem as the chemist, as the operator. So they should be running check standards, and it should be regular. If you ask about check standards and they have no idea what you're talking about, run to the hills, run to the hills. Really hoping the 3rd point's about to slide in. Hey, yes. Not the best with technology, so I had very low faith in that. May I see your most recent instrument calibrations? This one I'm going to go into more detail. I'm going to show you how to actually look at an instrument calibration and break it down. These are the standard of whether or not the lab is good. I'm going to go into this a little bit more. Dadgummit. No, this is right. Okay. Thought it was a different slide coming up. Okay. So there's a couple of things that you want to look for when you get one of these graphs. I am actually going to go to this. Let's evaluate one together. Da-da-da. All right. I do have this sourced, so if you go to the slides, you can pull this up. I literally picked this out, so it would be a bad example for what we're talking about. Do not get mad at this study. It's perfect for this study that I pulled this from, but it's a good example of a bad calibration curve for our purposes. Not the worst, but not the best, right? So we're looking at a few things here. What the heck is this? Don't let it intimidate you. This is one of those things that sometimes chemists will give you and try and intimidate you and try and act like it's really complicated. It's not. This is a line. That's it. That's all we're looking at here, right? There's 1, 2, 3, 4, 5 points, and they have made a line. Is there a laser pointer on this thing? That would be. Okay. So this R squared value right here is how linear is this line? How well does this fit a straight line? If it were a perfect line, that number would be 1, 100%. It's exactly a line. This is actually 99.68% a line. It's very, very high. When you're looking at these, this particular number needs to be 0.998 or above. It really should be, because what we're doing isn't hard. You can accept this, like for this study, this is fine. For this study that I actually pulled this from, a 996 is totally acceptable. But you really do want this to be 998 or above. You want it to be as linear as possible. The other thing is you want some kind of consistency or logic between these points because of the way the points are made. So when you buy a standard, it's 100 ppm, let's say. You then, as a manual person, have to dilute that to create your range. Because let me back up a little bit. The way this whole thing works is you have a detector and you have a solution of stuff. Let's say this solution of stuff is a mix of different cannabinoids. In between the two is a column. This column is charged. It has a specific charge, so polarity to it. And when you send the mix of cannabinoids through that column at a set rate, they're going to separate out based on their charge, because every molecule has a specific charge associated to it. So as they separate out, you're going to get different responses coming through the detector, right? The strength of that response is what we're setting up here. How well can it see the strength over a range, right? And so for this one, their range looks like maybe 3 to 45, perhaps, milligrams per kilogram. So that's the range that they've set up to see at. For us, what we typically see is 0.5 ppm up to 100 ppm. That's where most of our stuff sits. So when you see those, that's the range it is. All right. So you want to see linearity. That's one of the first things we're looking for, right? You want it to be a line. It's a little low. I would have some questions for this lab, for this 996, but it's not terrible. There we go. All right. So here I've pulled out the actual points that they've set up. 3, 7, 12, 25, 50. I hate that. And there's a reason, right? It's because when you buy that standard at 100 ppm, you have to manually, as the operator, create those dilutions. If you're going from 100 down to 50, that's an easy dilution. If you're going down from 50 to 25, that's an easy dilution. 25 to 12, that's not an easy dilution. 12 to 7, that's not an easy dilution. 7 to 3, not an easy dilution. There's some more complicated math. You want to see a pretty consistent logic to the dilutions because it is a manual person doing it. And if you have more math involved, there's more chance for error. So you want the math of dilution to make sense and be simple. Why make it more complicated on themselves, right? If they are choosing to make it more complicated on themselves with the intervals they've made, that's not the kind of technician you want doing your lab results. You want somebody who's getting rid of potentials for error, not creating new potentials for error. And then I don't see 6 calibration points here. For us and our purposes, we really should be running 6. This has 5. So 1, 2, 3, 4, 5 points that they have used to make their range. I really do recommend 6. It's not a requirement, but if you can remember these couple of things, is the line linear when they send you the results? Is there a consistent logical dilution interval? And are there 6 calibration points? This one's a little chemistry heavy, I will admit. But if you can remember those couple of things, you can pretty much know if the lab's calibration standards are good or not. The other thing you need to check for is the date. If they send you one of these and it's beautiful and perfect and you did all these 3 things, you're like, yeah, check, check, check. Date 2022. That is garbage and I've seen it. I have seen a lab give me a test result from 2-3 years ago, and that doesn't serve me. That doesn't do me any good. It needs to be happening. If it's a high volume lab and they're running lots of samples, it needs to happen every day. If it's a low volume lab and they're running 100 samples or less a day, they can probably get away with weekly. But if we're talking a high turnover lab, which a lot of these companies are now starting to be high turnover because of the proliferation of products, it needs to be pretty much every day. So a lot there. I tried to get through it quick because I'm trying to be mindful of the time. Any questions on that section on how to vet your third-party lab? Some questions that you might want to ask them.
Will a third-party lab offer up this information and serve them best to say, hey, this is our SOPs, how we go through it?
A good one should. A good one should. You shouldn't have to ask all these questions, right? But I say that a lot of them are stretched thin right now because they're so busy. So like, while they may be good operators and they may have the intent to want to give you all these things, they also know most people don't know to ask for them. And it almost becomes a waste of time because it does take a lot of time to gather these things and present them and get them back to you. But yeah, a good one should be giving you these things when you're initiating the relationship, when you're saying, hey, I need you to start testing some products for me. They should be giving you their calibration standards. This should be their sales pitch to you. Yes, this should not take them weeks and weeks to get. If it does, it should give you pause. Yeah, it should be very, very handily ready. Yeah, because again, if you cut these corners as a third-party lab, that's money. If you're not running those standards every day, those standards are expensive. So that is a big cost savings for you, right? So they are incentivized if nobody's watching them and there's nobody like, we really are in the land of we have to make sure that we're working with ethical chemists who are not going to cut those corners and are going to do what's right because they're not being held accountable. So they have to just be good operators. And that's where it's on us to stay on them too, right? Like I said, constant vigilance, you got to check them, right? Like just because you checked them once, check them again next year too. Check them every now and again. Some states do. So like if you are choosing to use a third-party lab that's in a state that has chosen to regulate the third-party labs, you're already starting at a little bit of an advantage because there's somebody holding them accountable. There's somebody going through and regulating them. But as I'm sure y'all have seen, like some of those labs that got vetted in California recently have come under scrutiny because it was looking like they might have been doctoring results to, yeah. So I mean, just because a lab is approved doesn't necessarily mean they're good. You know, so it is up to all of us as the operators right now to be on top of those labs and hold them accountable to make sure that we are being held accountable to our patients and are selling them the right stuff. All right. This is our cultivation team and some of our flower pictures from here in Texas. So that's a little plug there. But I do want to talk about this. This is one of my soapboxes that when I'm having beers with friends and they start asking me about cannabis, this is one of the soapboxes I get on. Natural occurring cannabinoids are CBG and CBGA. Those are your precursor molecules. They are kind of the parent molecules in the synthesis pathway that leads to pretty much everything else. It leads to THCA, it leads to CBD, THCV, CBN. All of those things originally start out as CBG. So if you have a plant that is harvested early, it's more likely to be higher in the CBG content. All right, we all pretty much well know about CBD and THC. Those naturally occur in the plant in their acidic forms. All that means, don't let people confuse you on that. That just means it has an additional functional group on it. There's another group of things added onto the molecule that make it acidic. Yes, sir. With what? Oh, no. So the guy in the front here, Dylan, that's our head cultivator. He grew that. Yeah. That's our grow here in Texas. Yeah, we're very proud of that. Hey, thank you, sir. We're trying the best we can. All right. And then I also see I threw some percentages up here for you because there's a lot of interesting, I'm not even going to call it good or bad, but there's a lot of interesting conversation about the percentages that occur in the plant naturally, right? You are not going to see a high amount of delta-9 THC in any cannabis plant anywhere, be it hemp or normal cannabis. You're going to see THCA not THC. It naturally occurs as THCA. That's why if you want it to work, you have to smoke it. You have to add heat. That's why if you've ever made an edible from plain flower and you didn't heat the flower up, you had no psychoactivity because THCA is not psychoactive. It's still medicinal. There's still medicinal value to THCA, but it is not psychoactive. So that's really important to remember. And I think most of the hemp folks know that because that's how we're selling flower here in Texas right now, is because a lot of plants don't have high THC content. A lot of them will be below 0.3, below 1% THC in the plant itself. Then we have some trace cannabinoids. There's a lot. There's a ton of these. Yes. Mm-hmm. Mm-hmm. It is Delta. That is Delta 9 in that instance, yes. And let's specifically talk about Delta 8 there too, because now we're getting into trace. You will see some trace Delta 8. And what I mean by trace is really low percentages, like 0.1, 0.2, even lower than that, right? So that's important to remember. It's important to keep in mind that it occurs in such small quantities that means something. And I'm going to get to that, right? It is. It absolutely is. And this is one of the things I'm going to be talking about. All of it's good stuff, isn't it? So now we're going to talk about synthetics. And I know that this is going to have an interesting and mixed feeling between all of us, right? But I'm going to say this. A cannabinoid is a cannabinoid. Right? Is it? It is. It actually is, right? So if you are getting a trace cannabinoid, let's use THCV as an example, and you're buying it in bulk and it's coming to you as a powder or a crystal, right? Is that okay? Was that derived from a plant? Probably not. It probably started that way, right? But this is why I was talking about trace, right? These trace ones down here. Trace molecules are that for a reason. You're not going to get a 5-gallon drum out of that by extracting it out of the plants. You're going to take CBD and you're going to synthesize it into that. That is what is happening. And we have all got to stop saying, especially the people that are making them, that they are extracting and isolating them from hemp. They're not. It's literally not possible at the quantities that they are supplying it out to all of the country. But that's okay. Synthetic isn't bad. It just means that you have to ask some more questions, right? It just means there's an extra couple of things that you have to do, because THCV from a plant and THCV that was successfully converted from CBD are both THCV. Now, we can talk about whole plant medicine and that and the value of that. I'm totally with you on that. That's a different discussion, right? But the molecule to molecule is the same. So you want to ask if you're buying any active cannabinoid from a hemp manufacturer. This is how you vet the hemp manufacturer. You want to make sure that they are testing for any solvents and processes used to create that molecule. Nobody is asking them these questions because they are telling you that it is naturally derived from the plant, because they have to in order to sell it. The FDA has said no synthetic cannabinoids. You can't do it. And everybody's going, oh, well, it's not synthetic. I just took the CBD and applied ethanol and heat, you know, and then they're telling you that that's natural. I swear to you that that is the case, because it cannot be. They cannot be selling you a 5-gallon drum of that extracted from the plant. There's not enough plants to do it, right? So you ask, are they testing for the solvents and the processes that they used to make it? Because and that's the question, because a lot of times the regulations haven't caught up to what they're doing. They might have created some novel synthesis that is using a solvent that the regulators are not requiring they test for. You want them to be testing for that, though, if they used it. So that's the question. Are you using a solvent and are you testing for it? Is there a process that you should be testing for? Would it pass under FDA or FTC guidelines currently? Pretty simple questions, but it is important to ask. Ice and water. You can do all naturals, yeah. Beware. I told you I was going to talk about HHC. I can't stand this one. This one scares me. What I've researched on it, it doesn't exist in cannabis that I've found. And if it does, it's so trace that you would really have to go looking for it. So it's not, if we're looking to the plant to guide us, which most of the industry does, we look to the plant, right? The plant is our guide. The plant says, I don't have very much of this molecule. Why are you trying to make so much of it? And how they make this molecule is they are just blasting it with hydrogens. So they are literally taking the molecule and covering it with as many hydrogens as they'll stick to it. And then it's HHC. I'm going to get into why that's bad. Okay. It's a novel molecule, first of all, right? And in this country, when you make a brand new molecule that's never been in people before, there is supposed to be a way to test that. You shouldn't just be guinea pigging that out, especially on people who are seeking medical care. That's not the population to test a new, I don't know what it is on, right? I love this slide. This is where oh crap, I'm running out of time. So I will go a little quicker so I give you all time to talk. This is an electrostatic graph of THC and CBD, right? When I was talking about them going through the column and having polarity, this is a graph of what that polarity looks like, right? You've got concentrations of polarity, positive and negative charges on the molecules, right? These interact with your receptors in your body based on that shape and based on that polarity. So if you blast this with hydrogen atoms, you have now put a positive charge all over this molecule. Will this look the same if you stick positive ions all over this? No, it's going to be a different shape and it's going to have different charge. So its ability to go into your receptors and act is altered. It may still fit and work within the endocannabinoid system, but you've done something to it now that is not natural. So you do need to be really careful that if you're using a cannabinoid, make sure it occurs in the plant. Do some research and double check and make sure that it's actually occurring in cannabis. Also beware of bad synthesis. I've seen some products on the shelves that were literally like THCV, CBG, CBD. It was like 12 different trace cannabinoids in there. That is the definition of a bad chemical synthesis. A good chemical synthesis is one output, right? If you just get a synthesis of a bunch of random molecules and you're like, I don't care, ship it and sell it. Do beware of those kinds of products. Okay, transparency. Running out of time, so I'm having to go a little bit quicker. Oh yeah, and Terrence, for those who aren't aware, is here in the front row. We've got to give patient access to COAs. It's huge. Once you've established that good relationship and you've done all that really hard work to make sure that it's all correct, be loud about it. Don't sit on it. Like, actually put it on your website. Put it out there to your patients. Make sure they know that you're doing all those things to take care of them. You've got to educate them on what your label claim is, what that variance is that we talked about. Patients have no idea that their ibuprofen is a variance, right? So sometimes we're teaching them that that is normal, that products do vary. You got to watch out for the synthetics. You got to make sure that they're natural, right? Like it does occur in the plant. There's a couple of questions to ask with synthetics, and you got to make sure that your doses are consistent throughout. This last one is a little pipe dream. I've run out of time. I want to give you all time to ask questions, so I'm going to skip that. But basically what that is that's saying we have to do it ourselves. We cannot wait on the government here, y'all. Like if we wait on the government to regulate us properly, we're all going to be dead and gone. Like it's just not going to happen. So I think that if enough of us all got together, and it doesn't even have to be something that's a paid or anybody's making any money off of it. It could literally just be a coalition of all of us getting together and saying, if you're in this coalition, you choose to operate by these rules, this variance, this logo, these things. And if we could all do that, at least out of all of the chaos that's currently existing right now, we could say these operators, Patients of Texas, you can trust it. If it's got that sticker on the door, come on in. You know that they're good. It's not gas station weed, right? I made it. Any questions? Please feel free. I'm very, very open. I will answer, even if it's on or off topic of the things that we've covered. I'm more than happy to answer questions. I personally think it's going to be FTC, especially in the hemp space. I don't feel like the Farm Bill folks are going to get slapped with FDA requirements when we get federal legality. It's most likely going to be the FTC. And it's similar. The main difference between those is that variance allowance. But the label requirements are pretty much the same.
What's that?
That's what we were talking about, right? It's usually like 10% versus 20. Yeah, 15 is a good rule of thumb. Yeah. Because again, if it's going to be the FTC, it's probably going to be more like a 20. And if it's the FDA, it would be closer to 10 based on the other drugs that are in the market currently. Yeah, we actually, through the medical program, we sell a salve that actually won a High Times Cannabis Cup Award up in Massachusetts. So we're pretty proud of that one. But yeah, you can do, there's so many different things you can do. You can do topicals, patches, capsules. Truly, it is up to your creativity. Like if you want to sell a product, I mean, I remember I had somebody pitch me back years ago, like a Listerine tab, right? They had figured out a way to do like a breath strip. I've seen mints, yeah. So if you want to make it, you can figure it out. There's a way to do it. Even the water, honestly, water used to be the big toughie, right? But man, go on DoorDash. Yeah, you can go on DoorDash and you can get it straight to your door. Yeah, cans of water. Yes, ma'am. So no, I will tell you too, you are in a good position through the Farm Bill in that seeds are legal through the Farm Bill because that sounds like a good contact for you. But I will say seeds, you can pretty much get shipped over from anywhere. Oh, I would put you really shouldn't run into that. If anybody, like for instance, you could even say like that you were pheno hunting. And so say you got your seeds and you're growing them out and now you're sending them off for testing and one of them comes back hot for THC, heaven forbid, right? It's not a big deal, right? Like you just say, oh, okay, thank you, I'll cull those. That's the pheno that I'll cull, whether or not you do, up to you. So but you're not restricted to only grow. Beats me. That's probably a get you started, but I wouldn't. Oh no, I fully believe that. There's some of my regs that tell me like what things to test for. It's just the wrong reg referenced. Yeah, because even if you started with, like she said, if you start with strains from that, 6 months from now, they're not that anymore. So yeah, you shouldn't, don't worry about it. And again, even worst case, it comes back hot. It's not a big deal. You just call those and you move on, right? You just say, okay, I knew which plants, which table that was or whatever. They're gone. Thank you. Yeah, of course.
You can Google Grower's Choice right here in Texas and have taken each strain you want, seeds.
There you go.
And they'll send it to your front door to your house.
No, I've already ordered.
Yeah.
Grower's Choice.
Yeah.
All right, I'll put it in.
And it's legal.
Yeah.
Have you that?
Yes, ma'am.
It's no weight. That's why it's legal. No weight on the seed. Buy all the seeds you want.
Yeah. I definitely would be curious if they're new. Like if they're a brand new startup lab, there's going to be some more questions, right? If they're already a fully functional operating cannabis operator and they're testing for, they can give you a list of all their people. Now, granted, don't let that be the end all, right? It's a good, like, okay, check. They're testing for a lot of people. It's one of the things. But no, I mean, the things that I covered are really the big ones, like that check standards, check the calibration curve, and those things that we went over in the calibration curve. And then, yeah, probably their size, because that's another part of it. Like from a cost standpoint, you do want to check about their turnaround times. That's going to be potentially detrimental to your manufacturing. If they tell you we're so busy, we're not going to get you turnaround times for 4 weeks, bye. I can't have that, right? I can't have stuff sitting on the shelf for 4 weeks waiting to move, right? Waiting to know if it passes or not. So that's a big one, turnaround times. Do you want to know turnaround times? And you want to know the cost because you're going to be using them a lot and you don't want to be scared to do another test. You want it to be a good relation and you want to double check on those retests and what they're charging for those retests because they should not be at the full price. Because a lot of times they can just rerun that same dilution.
Okay. What's the variation in pricing for our taking?
All over the place because you can, if you're in the hemp space, you have, call it the plus or the minus, you can go over state lines and you can ship your products over state lines. It's going to add time, But if the lab is faster or better, it might actually be worth it to ship it out of state. But that also comes with its own risks, right? Like if you're shipping a gummy, is it going to survive? If you're shipping a chocolate bar, is it going to make it? Does the lab care if it shows up whole or can it be melted? You know, all those sorts of things. It really does depend on what you're sending them. But yeah, the big things are the things I highlighted. We do. Yes. That's my background, actually. We do. Yes. I helped develop. Yes. Yes. We only test it's an internal lab, so it's our own in-house, and then we do external labs to validate. So we're not testing for other folks, but we could get into that. I thank you. I try to think so. I agree. No, I agree. It's a big hole in the space here for sure. It is hard. I'll tell you, if I had a good chunk of capital, I'd be doing it. I've seen one or two that I've evaluated because when we were going through, when the DPS was trying to look at using a third-party lab to test us against, I was recommending some in-state labs. They ended up going with an out-of-state lab, much to my chagrin. I'm not going to say a name today because I don't want to endorse anybody, but y'all have my contact info today. Yeah, there are some around. And I was going to say, y'all can take my contact info today. If y'all need help vetting a lab, that for me doesn't take.
Yeah, I'm curious what labs are going to look like. You know, because there's so many that are already around, right? That you can already go to. Because Texas is what it is right now. And there comes that is that for people to.
It will be.
So I talked to one, I scouted on the lab, I don't list of the pretty much getting them to respond was kind of an issue, but I didn't know. It was really weird to get people to call me back and tell people already, so yeah.
They kind of look down their noses at us.
Then I would ask them for I would ask like that's the guy, my cordial, and that, and they'd be like, oh, well, let me check. And they would send that out to like Oregon. So that they would do like really the basic kind of like getting it out to another state, Oregon or California, and then they can do that. And so I did find one blue bonnet out of Dallas. They never clearly used me, but I asked them those questions, you know.
Yeah.
I was like, I'm good science, but they actually told me that when they first got in the industry, like Sid Miller was really pushing like the hemp market out here and stuff. And what happened was like go a bunch of lab teams set up and we're like, yeah, we're gonna have a big business. And then don't get them like this is a lot of them out in Mexico as well. Yeah,
I think what will tip it.
oh yeah, they were bringing their capitals for us.
Oh, yeah.
I think what will tip it in the direction that's really going to make it happen for Texas is a requirement. Right now, no one except the medical is required to test every batch. Once everybody is required to test regularly, now that regular influx for those businesses is there. That's what happened in Florida, right? Like that's where I originally started. Oh yeah. To what levels, to what variances? Yeah. Well, and even then, you've got to, it's the constant vigilance thing, right? Like I said, there was some scandals recently that came out of those labs in California. So even the labs that go through all the scrutiny and have all the regulatory oversight, sometimes, yeah, so you as the operator have to be the one. Again, because we can't wait on the government. We can't trust the labs. We have to be the ones that's making it correctly. No, you're good. No. Yeah.
One of the things that we talk about, like with this other stuff, when they're making it into other things, it is producing and it's created. So yeah, it's consistent.
Yes. Yeah, so the short answer is yes, and I will try not to go too chemically to this answer, but just from a synthesis standpoint, there's stuff called metal catalysis, right? And that's where you have a metal involved in the actual synthesis. It requires as attachment to that metal surface. Often those are heavy metals. So yes, absolutely, there are some synthesis that could be causing heavy metal contamination. That's why when I'm saying like ask those people, ask them what their processes are, because usually heavy metals are required to test for, but it's a list, right? And we've all just talked about those lists are garbage and they were made by legislators, not chemists. So they may have missed one of them that they're testing for, one of them that they're using for that catalysis. And they're like, oh, of course it always passes because I'm never having to test for 10 because I'm not required to, right? Absolutely. Yeah, for sure. We're constantly trying to offer more things within the confines that we're currently allowed to do. Constantly looking for clever ways to maybe read the rules to try and sell something. Transdermals are actually pretty low lift in that regard. We've also, some of those products, unfortunately, because of the patient awareness, they just don't take off, right? So like we did suppositories for a minute. Great product. I love suppositories for cancer patients, for epileptics, for menstruation cycles. There's so many positive benefits to them. But because in general, suppositories are one of your lower movers in your product categories, if you don't have enough patients, your low moving stuff like transdermals and suppositories, they're more of a lift than the squeeze is worth oftentimes, which sucks. I love those products. Yeah, and it truly is one of those things that like with those kind of more niche products, when the program kind of hits a critical point where we have enough patients, suddenly we can start offering those more kind of niche, cool products that are a little bit more heavy and more medical, honestly. A transdermal is a more medical product. It's a very, you're not going to a festival and slapping patches on yourself, right? Like, no, I miss the suppository. I think that's like, for me, that's kind of the transdermal for us that we tried. And we tried it out and it truly was just people who liked it, loved it, but they were few and far between. Yeah. Right. Uh-huh. Yep. No, you're totally right. You're totally right. But that particular patient population, we'll call them, I think is a little bit more willing to try things than the average bear. Yeah, that's your guinea pigs right there. Yeah. Oh God. Yeah. No, I'll talk about that. Yeah. No, so Terrence just asked me about mobile testing kits. Y'all might have heard of Orange Photonics. I wasn't going to say their name and crap on them specifically, but I will. Those don't work. Just point blank. No offense. The mobile testing units, the little. Yeah, I've got those too.
Then my grower really put cash on it. So I'm like,
Yeah, we had one. We've validated against it. Let me tell you, if they worked, cops everywhere would have them in the back of their thing tomorrow. So that is your biggest indicator to know that those do not work. Because yeah, I mean, it's a literal, like if the idea worked, it's a briefcase size HPLC that literally is less than a minute of prep. A cop could have one of those in the back, test you on the side of the road and know right then. I'm sorry. There's no way. Yeah. Oh yeah, thanks. Thanks y'all, appreciate it. References if you need them, slides if you want them, reach out.